What's up, bio students? This is section 2 on cell division. Kind of a beefy unit or beefy section. I'm going to try and go through it as quickly as possible. But we're kind of encompassing two things: the cell cycle, as well as what's happening in this mitosis phase, which is actually a lot. So we'll go through these relatively quickly, but spend a lot of time talking about that. So a little bit on section 1. You don't really need to write this down, but I just want to kind of refresh your memory as to kind of why cells divide. We're here to increase an organism's surface area. You guys did a lab on this yesterday, or maybe today, or maybe Monday, whatever day it would be. But you'll notice that plant cells will differentiate and grow into stems, roots, and leaves. Our cells are going to differentiate into skin, muscle, bone, nerve. So a lot of things that occur in this cell cycle are the same for each cell, but when we get to a certain point, we will differentiate into things that we need, whether it's replacing old cells or kind of creating new ones. So the question I'm going to ask is why do we need the cell cycle? Why do we need to go through this process? Well, we need to regenerate cells. That's an obvious one. You look at skin cells, on average, they're going to probably go through about a month of life and then die and then be shed off. So you need to have new ones growing. Whereas nerve cells, you're born with what you got. So whatever you have for nerve cells, if you kill any, they actually do not go through this process. So it's kind of important with skin. Unfortunately, it doesn't happen in nerve cells. But one example of where, if we do not have any regulation, what can happen, and that's cancer. Cancer is basically uncontrolled cell division. That's where we generate, massive amounts of cancerous cells, whether it be tumors that are, growing in various parts of the body, on various organs, and then metastasizing, which is a big fancy word for spreading throughout the body. But as you can see, it's honestly uncontrolled cell division. And what ends up happening is we're constantly creating these new unhealthy cells where your body is trying to attack them and kill them, and sometimes it just can't because it's kind of out of control. The other thing I want you to remember is that we need this cell cycle. We need to use the old cell as a template. And that's really, really important. So in a normal healthy cell, if you want to recreate one, you kind of got to throw it through the copy machine and go through this process. And that's what we're going to talk about today. So one thing that we're going to discuss is the simple breakdown. When we take one single cell, we're going to call that a parent cell. And we're going to make a copy of that parent cell. And to do that, we are going to first break down what we classify as the nuclear membrane, which kind of surrounds the cell, okay? Or not the cell, the nucleus. So if this is your cell, this is your nucleus. The nuclear membrane, as you can see, is breaking apart. That information, that DNA, that blueprint, that codes for basically all the information we need to make that cell is normally in a form called chromatin, and it's actually going to be condensed into chromosomes at this point. Those are going to separate and become visible to allow for replication. So one of the first things we're going to do is to replicate, and that means to copy. So if you're going to make a new cell, you better copy the instructions to do that, because not only do you need those, but that new cell is eventually going to need to be copied into another new cell. In order to do that, got to have that DNA. So let's take a look at the big picture here. What are the major steps of the cell cycle? This big gray interior, so starting at this blue line, we're going to say this is our starting point, all the way up to this purple, sure. That's considered interphase. Inter means between. These are the between phases before we actually split and divide. So the mitosis and cytokinesis is where we have a brand new cell being made and split. Through this process, we're going to do things like grow and replicate and copy and begin to prepare to make a new cell. Timelines will vary, and we'll discuss those, but big picture, here's a cell. We're going to grow, copy, get prepared to break into two new cells. So what are some of the things that are going on in interphase? Interphase is the period of time before a cell will divide. So individual chromosomes are not visible at this point. DNA is replicated or copied. Eukaryotic cells are always going to be in one of the 5 phases. So either you have a cell in your body that's in G1, S, G2, M, or about to go through cytokinesis. In human cells, this lasts an average of 12 to 24 hours. So to take a cell, to grow, copy, prepare, divide into 2 cells, we're looking at about a day. But imagine this happening, exponentially in some cases, not always. In certain parts of our body, like skin cells it is, we have multiple cells going through this at once. So I don't want you to think that this is just one cell in your body. Every cell is always in one of these 5 phases. So let's start with G1. G1 or gap 1 is where this process starts. And there's some big things that I want you to know. So let's say this is a brand new cell. Just got copied, and it's about to go through its process of life. It is going to begin by growing, okay? It is going to take on nutrients, grow in size. It's also going to synthesize or make RNA, proteins, and other macromolecules to kind of get bigger, get stronger, and get prepared. There is a checkpoint right here. It's called the G1 checkpoint as we're going through this. And, that's basically to say, hey, are you big enough? Are you tall enough? I like to consider it like when you go to a roller coaster and it's like, are you tall enough to ride this ride? If not, you're going to go back through this process and continue to grow. If so, if you meet the requirements, welcome to the S phase. So, next on the list, I just want to talk about the fact that sometimes these cells will take a little bit of vacation. And I've talked about G1 here and how you got to grow and meet this checkpoint, but sometimes you'll float out here in something called G0. Most cells in adult multicellular organisms are in G0, and it's a phase where they're metabolically active, which means they're alive, but they're just not dividing. There might not be a need, a necessity. This might be like a liver cell, or a muscle cell that you just don't need to do anything with it yet, so it's not going to prepare itself to go through this process. Just going to kind of hang out, and we'll classify as a holding pattern. So, if you get a cell and they're done with their little vacation in G0 and they've grown and they've met that checkpoint, well, welcome to S phase. S phase means you've passed the point of no return, which means that after this checkpoint, if you are tall enough to ride the ride, we're going to divide. So the S phase consists of 2 simple things. One, DNA replication, where we copy the genetic information. As you can see, S means to synthesize or to make, so we are going to copy that information. And what this is going to do is it's going to double the number of genes inside the nucleus. So that's like saying your DNA copied itself, and now you have 2 blueprints inside one cell, inside one nucleus. Next up, we have the G2 phase. So again, as we've copied that DNA, we are now into G2, and that's kind of a prep phase. More growth, cells got to get bigger, because remember, you're taking one cell and making it into 2. You have to make the plasma membrane bigger. You have to make sure organelles are bigger. So you're going to synthesize more proteins. These proteins are going to help make Golgi apparatuses, and they're going to help make ERs, and they're going to help make ribosomes. All the necessary things that you've learned about inside of a cell, we're going to copy enough of to make another cell. So it's basically we're fitting, 8 people into a 4-passenger car. One way to look at it. There is a checkpoint here at G2. Once it reaches that G2 checkpoint, it will determine if it is ready to divide into the mitosis phase. So a couple of things to talk about in the M phase before we go through it. At this point, cell growth and protein production has stopped. The cell's energy is focused completely on making another cell. So we're right here. All right, we're right here where it says prophase. And as in both G1 and G2, there is a final checkpoint, which is going to be right here before prophase, and right before metaphase, which basically says, hey, are we ready? Are we sure? That we're going to divide, or do we need to hang out for a little bit more? It's going to happen, but maybe we're just not quite ready. So we're going to kind of go into that and explain how we go through this M phase. So far we've taken our vacation, we've grown, we've replicated DNA, we've grown a little bit more. We're preparing to actually make 2 cells. When that M phase is complete, just to kind of go through it from a bigger picture, the nucleus will split into 2 new daughter cells. So as you can see here, we'll talk about that. This is the goal. Mitosis means we are going to make 2 cells. So that is the goal. We're going to take one and make it into another, and that we have the old one and a new one, and you are ready to rock. Some examples of cells that go through this, and I kind of mentioned it before, so I'm going to gloss over it quickly. Bone cells are constantly dividing. Skin cells are constantly dividing. I talked a little bit about your liver cells. They're usually in G0 just because they don't need to be replaced very often. And then nerve cells, and it says rarely, never divide. I mean, if you damage your brain or spinal cord, it can't be repaired. So let's talk about this mitosis. First one, and it kind of goes in this PMAT, P-M-A-T. Prophase is first. Things you need to know. Well, number one, I would jot a picture down, to be quite honest, for each of these slides. It is the first step of mitosis after G2 is completed. Chromosomes become visible as paired chromatids. The nuclear envelope disappears. So what it means is, hey, we got chromosomes, we got a nuclear membrane breaking apart, or envelope. Next phase, metaphase, second step, mitosis. We have these centrioles at the end, and they are going to basically reach out with these spindle fibers and attach to the centromere. Centromere is like a little dot here that brings these two sides of the chromatids together, and it's just going to attach. But the other thing that you need to realize besides the attachment is notice that all of these line up on this, what we classify as the metaphyseal plate, okay, which is right down the midline of the cell. That way, in a moment, you'll see that these are going to break apart into two. Step 3 is called anaphase. So prophase, metaphase, anaphase are third step. Spindle fibers pull the chromatids apart, bringing half of them to each side of the cell, as you can see. And opposite poles of the cell now are containing half, of each chromosome, which is really giving it information on both sides. And we're kind of, again, preparing for the split. Our next phase, and it's really the fourth step of mitosis. A lot of people say it's the final, but I don't consider it. Telophase is your fourth step. Nuclear envelope begins to reappear. You can kind of see it as around this outside here. Around duplicated chromatids. Remember, this information was copied in that cell cycle. So whatever you have here is the same information here. Cell membrane begins to fold in, as you can see at this location, and we are getting close to breaking apart into two cells. Last but not least, we have cytokinesis, and that's what ends mitosis. The cell membrane will literally cut in half, and now you have two cells with duplicated genetic information. Notice that our chromosomes have unwound. All of our DNA is in chromatin form, and cytokinesis has basically finished this process. So a cell that is freshly born is either going to be in G0 or G1. From there, it moves to S, to G2, then through all four steps of mitosis, which is prophase, metaphase, anaphase, telophase, finally cytokinesis. So here are some questions to ask yourself, as I always do. I'm not going to read them off to you, but if you learn anything in this video, or if you're kind of going back through your notes, these are the types of questions you should be asking yourself. Big things to understand is chronological order, starting out with what, ending with what, and making sure you understand what's happening in each process. Other than that, I think you guys should be ready to rock. If you have any questions, talk to your local biology teacher. Other than that, that's all I got. Yago out.